Yeah, so that's basically anything we can eat or drink in the lab. Thanks, Oksana. So as Oksana said, we've used DNA methods to look at a range of issues, mainly B viruses and pathogens such as Nosema, more recently AFB, but in the last year we've been looking at developing a new assay for resistance mutations to the synthetic pyrethroids that some of you may have heard George from Bayer speak about just before lunch. And we'll see here if we've got a pointer we have. So this here is a voltage-gated sodium channel. And it's basically a protein that crosses the membrane. And what it does is it basically transmits nerve signals in Varroa. So it's the target gene in Varroa for the synthetic pyrethroids, that is for Apistan and Baverol. And as I said, it prevents the nerve signals, which leads to paralysis and death in the Varroa. The way it works, it's a bit like a lock and a key. The lock is the sodium channel there. The key is the pyrethroid. The pyrethroid, like a key, will bind into the sodium channel, block it, leading to Varroa death. This particular point here, which has the star in it, is the binding site for the pyrethroids. So it's a very localized binding of the pyrethroid. And there are 3 specific DNA mutations, 3 DNA changes that lead to resistance. And these 3 DNA changes basically change that lock, if you like. It's like knocking out the pins of the lock. The key is no longer going to fit. Okay, so whether you add more strips or try and jam that key in harder, it's not going to fit in the lock. The voltage, the sodium channel will still transmit, the Varroa will still live. That is, we have resistance. So the 3 DNA changes leading to resistance, we have one described in 2013 in Europe, and in fact has very recently been described, it's now worldwide, called the 925V. And this is just describing an amino acid change. I'll show this in a bit more detail shortly. The same amino acid change, a different DNA letter changing in the United States in 2015, and another change of that same amino acid to L925I, also at the same time as the L925M. What that actually looks like from a DNA level, which is where we like to play, is this. If we unwind this into a sort of DNA sequence along here, here's the 3 letters that make up the amino acid. So 3 letters of DNA to an amino acid, it's strings of amino acids that make up a protein. So the 3 mutations that I've just described there, that C changes to a G, and that's the L925V change. That same letter changes to an A. That's the L925M mutation. And for the L925I, we get an extra mutation, that same amino acid. So it's what we call a hotspot. It's a very localized target change in the DNA sequence. So the methods to detect mutations, the method that George spoke about, where they did it, I think at a Bayer laboratory or in Spain, is using a whole lot of synthetic DNA probes for each different mutation. For us, as this was sort of work privately funded by us, essentially, it was about $4,000 to set up. And so we weren't going to spend that money on it. So we decided to look at another method. And this is a method that we've used quite a bit in our laboratory. And it's a version of the method that, for those that remember Peter and his team describing the CSD allele work, which is looking at DNA melting behavior. And so this is called Snapback high-resolution melting analysis. And basically, the message from this to take is basically a form of qPCR that we're hearing about more and more for Manuka or for AFB. So this is a form of qPCR. It's essentially just using 2 DNA primers here for PCR, but we have an extra element on one of the primers. And I'll show you what this looks like. It means that when we do the melting of the DNA sequence, we get 2 bits of DNA to look at. We get the whole amplicon region, so spanning the DNA sequence, and a much more localized one where we can look at those 3 mutations. So what it looked like for us with the snapback genotyping, we could scan that whole region, not just that amino acid, but about 4 or 5 amino acids around that mutation hotspot, and look for changes. So here is the normal sequence. So each line essentially represents 1 Varroa DNA sample. Unfortunately, the L925V melts identically, and we knew that because we'd mathematically modeled this, and so we knew that was going to be an issue. But quite separately, we can see the L925M and the L925I mutations. So quite clearly differentiated. But in the same reaction, we also have that, what we call the snapback element. And what that allows us to do in that same reaction, there's a bit of DNA where we looked at the changes. We now see these 1, 2, 3 different peaks. And so there is the lines representing the sensitive Varroa samples, the lines representing the L925V samples, so we can clearly differentiate them, and the L925I samples. So essentially using just 2 PCR primers in the reaction, we can differentiate all 3 mutations. So the results, we coupled this new PCR method with a high throughput method for Varroa DNA samples. Dozens of Varroa, in fact over 100 individually tested. As George said, none of the 3 mutations were seen, and we confirmed this by DNA sequencing of that region. So, what's at play? Do we have mite bomb hives that we've heard about where we have low numbers of Varroa in various hives, and a particular hive or hives in an apiary will have massively high numbers? Or is there another resistance mutation that the test is not currently seeing? And that's something we plan to investigate in the future. Thanks very much.